structure, and function of the neurologic system. The nervous system is divided into two main parts: the central nervous system, or CNS, and the peripheral nervous system, or PNS. The CNS consists of the brain and spinal cord. The PNS includes cranial nerves and spinal nerves that branch out to the rest of the body. Information travels through afferent, or ascending pathways, and efferent, or descending pathways. Functionally, the PNS is split into two systems. The somatic nervous system controls voluntary movements like skeletal muscle activity. The autonomic nervous system regulates involuntary body functions such as heart rate and digestion. It includes both sympathetic and parasympathetic divisions, which work in opposition to maintain internal balance. The nervous system is composed of two main cell types: neurons and neuroglial cells. Neurons are the primary signaling units that transmit electrical and chemical signals. Neuroglial cells support, protect, and nourish neurons, and they also help facilitate neurotransmission. Together, these cells allow the nervous system to function efficiently. Neurons vary in size and shape, but they all share essential components. Inside each neuron are microtubules, neurofibrils, micelle substances, and microfilaments. These structures help with internal transport and maintaining the neuron's shape. Despite their differences, all neurons are built for rapid communication. Neurons have three key structural parts: the cell body, dendrites, and axon. The cell body, or soma, contains the nucleus and is usually located in the CNS. Dendrites extend from the cell body and receive incoming signals. In the PNS, groups of neuron cell bodies are called ganglia. This diagram likely illustrates the anatomy of a neuron, including the cell body, dendrites, and axon. These components work together to receive, integrate, and transmit information. Structural features of neurons help determine how signals are processed and relayed. The axon is the part of the neuron that carries electrical impulses away from the cell body. The axon begins at the axon hillock, a cone-shaped area where signals are initiated. The initial segment has the lowest threshold, making it the usual site of action potential generation. This ensures efficient signal transmission down the nerve fiber. Axons are often wrapped in a myelin sheath, a fatty insulating layer that speeds up signal conduction. Nodes of Ranvier are gaps in the myelin that allow ions to jump from one segment to the next, a process called saltatory conduction. Axons end in synaptic terminals, where signals are passed to the next cell. Neurons can be classified by the number of processes extending from the cell body. Unipolar neurons have a single process. Pseudounipolar neurons appear to have one process that splits into two branches. Bipolar neurons have one axon and one dendrite, while multipolar neurons have multiple processes and are the most common type in the CNS. The diagrams show examples of each neuron type. These structural differences allow neurons to perform specialized functions depending on their location in the nervous system. For example, sensory neurons are often pseudounipolar, while motor neurons tend to be multipolar. Neurons can also be classified by their function. Sensory neurons transmit impulses from peripheral sensory receptors toward the central nervous system. Associational neurons, or interneurons, connect neurons to other neurons within the CNS. Motor neurons carry impulses from the CNS to muscles or glands, producing a response. Neuroglia, often called nerve glue, make up about half of the total brain and spinal cord volume. These support cells are found in both the CNS and the PNS. They play critical roles in maintaining the environment around neurons, providing protection, support, and nutritional health, and assisting in repair after injury. In the CNS, There are several types of neuroglial cells. Astrocytes help maintain the blood-brain barrier and regulate nutrients. Oligodendrocytes produce the myelin sheath around CNS neurons. Ependymal cells line the ventricles and produce cerebrospinal fluid, while microglia act as immune defense cells within the CNS. In the PNS, the main types of neuroglia are Schwann cells and satellite cells. Schwann cells form the myelin sheath around peripheral nerves, while non-myelinating Schwann cells support unmyelinated axons. Satellite glial cells surround neuron cell bodies in ganglia, providing physical support and regulating the environment. Mature nerve cells do not divide, meaning injuries to neurons often result in permanent functional loss. In cases of axonal injury, Wallerian degeneration occurs. The portion of the axon distal to the injury degenerates, myelin disintegrates, and the axon breaks apart. Swelling and hypertrophy of neurofilaments are common features of this process. Proximal to the injury site, the cell body responds by increasing protein and mitochondrial activity, attempting to regenerate the axon. New sprouts may emerge from the proximal stump. However, regeneration primarily occurs in the peripheral nervous system, not the CNS, and success depends on the injury's location, the extent of inflammation, and scar formation. This visual highlights the differences between healthy axons and those undergoing Wallerian degeneration. The figure shows the breakdown of the axon and myelin sheath, and the swelling of the cell body as regeneration efforts begin. Understanding this process is key for therapies targeting nerve repair. Neurons communicate by generating and conducting electrical and chemical impulses. They selectively alter the electrical potential across their plasma membranes to initiate an action potential. Once triggered, the action potential follows an all or none principle, meaning it either fires completely or not at all. Neurotransmitters are then released to influence nearby neurons. A synapse is the junction between two neurons where communication occurs. Signals are transmitted across the synapse either chemically, through neurotransmitter release, or electrically, via ion channels. The presynaptic neuron delivers the signal, while the postsynaptic neuron receives it. Synaptic boutons and synaptic clefts are critical structures for this transmission. The visual highlights how neurotransmitters are released from vesicles at the presynaptic terminal into the synaptic cleft. They then bind to receptors on the postsynaptic neuron, triggering a new electrical impulse. Proper synaptic transmission is essential for everything from muscle movement to complex thought processes. Neurotransmitters are chemicals that allow neurons to communicate by opening or closing ion channels. Some neurotransmitters are excitatory, promoting the generation of action potentials, while others are inhibitory, preventing them. Summation of signals can occur over time, known as temporal summation, or across different spaces, known as spatial summation. Balance between excitation and inhibition is vital for normal brain function. The brain contains millions of neurons and is responsible for reasoning, mood, intellectual function, and motor control. Major components include the cerebrum, which makes up the largest part of the brain, the forebrain, midbrain, and hindbrain, and the brainstem, which includes the midbrain, pons, and medulla. Each part plays a specialized role in coordinating body systems. Visuals here highlight the complex structure of the brain, including its lobes and subdivisions. Detailed diagrams show how different areas are specialized for functions like movement, sensation, vision, and language. Understanding brain anatomy lays the foundation for diagnosing and treating neurological disorders. The reticular formation is a network of interconnected nuclei found throughout the brainstem. It plays a crucial role in regulating vital reflexes such as cardiovascular and respiratory function. Together with the cerebral cortex, It forms the reticular activating system, which is responsible for maintaining wakefulness and consciousness. The telencephalon includes the cerebral cortex and basal ganglia. The cerebral cortex is composed of gray matter and is the center for conscious thought and voluntary movement. The basal ganglia control motor function and are composed of structures like the caudate nucleus, putamen, and globus pallidus. The limbic system, including the amygdala and hippocampus, regulates emotion and memory. Here you can see the basal ganglia and limbic system visually represented. These structures are located deep within the brain and are essential for fine motor control and emotional regulation. Damage to these areas can lead to movement disorders or emotional disturbances. The cerebral cortex is divided into 4 lobes, each with specialized functions. The frontal lobe controls reasoning, planning, and voluntary movement. The parietal lobe processes sensory information. The occipital lobe is dedicated to vision, and the temporal lobe manages hearing and memory. Together, these lobes create the foundation of human behavior and perception. The diencephalon is composed of several important structures. The epithalamus helps connect the limbic system to other brain areas. The thalamus relays sensory information to the cortex. The hypothalamus regulates homeostasis, including body temperature and hormone release. The subthalamus assists with motor control. Each plays a vital role in maintaining bodily and emotional balance. The midbrain, also known as the mesencephalon, acts as a vital relay center for visual, auditory, and motor system information. It includes the corpora quadrigemina, with superior colliculi for vision and inferior colliculi for hearing. Other important structures are the tegmentum, which contains the red nucleus and substantia nigra, and the basis pedunculi, which carries motor fibers. The midbrain is essential for sensory motor integration and reflexes. The hindbrain consists of the metencephalon and myelencephalon. The cerebellum controls fine motor skills, balance, and posture. The pons serves as a bridge between the cerebrum and cerebellum and assists in breathing regulation. The medulla oblongata controls heart rate, respiration, blood pressure, and other autonomic functions. Together, these structures are critical for survival and coordination. The spinal cord lies within the vertebral canal and is protected by the vertebral column. It acts as the communication highway between the brain and the body, conducting both motor and sensory signals. The spinal cord also manages reflex activity and basic motor pattern control. It ends in the conus medullaris, with nerves continuing as the cauda equina. The spinal cord is covered by 3 protective layers called meninges: the dura mater, arachnoid mater, and pia mater. Gray matter in the spinal cord contains the dorsal horn, lateral horn, and ventral horn, each with specific functions. Surrounding the gray matter is white matter, composed of ascending and descending tracts that transmit information to and from the brain. The illustration likely shows a cross-section of the spinal cord, highlighting gray matter and white matter. Gray matter appears as a butterfly-shaped region in the center, while white matter surrounds it. These areas are organized to efficiently relay motor and sensory information. You'll see detailed pathways for sensory and motor tracts. Damage to different areas of the spinal cord can cause specific sensory losses or paralysis depending on which tracts are affected. Understanding spinal cord structure is essential for clinical diagnosis. The reflex arc is the simplest nerve pathway and is responsible for reflex actions. It requires a receptor to detect the stimulus, an afferent sensory neuron to carry the impulse, an efferent motor neuron, and an effector like a muscle. Reflex arcs allow rapid, automatic responses without direct involvement of the brain. The diagram shows how sensory input triggers a rapid motor response via the spinal cord. Reflex arcs help protect the body from harm by generating immediate action. Examples include the knee-jerk reflex and withdrawal from a painful stimulus. Motor neurons are divided into upper motor neurons and lower motor neurons. Upper motor neurons are entirely within the CNS and control voluntary movement and reflex modulation. They synapse with lower motor neurons, which originate in the spinal cord and directly influence skeletal muscle. Damage to upper motor neurons leads to spasticity, while damage to lower motor neurons causes weakness or paralysis. Lower motor neurons form motor units by innervating one or more muscle fibers. The connection point between a lower motor neuron and a muscle cell is called the neuromuscular junction. This is where acetylcholine is released to trigger muscle contraction. Efficient function at the neuromuscular junction is crucial for voluntary movement. This slide focuses on the neuromuscular junction, the critical point where motor neuron axons meet muscle cells. It's here that electrical signals are converted into chemical signals using neurotransmitters like acetylcholine. This causes the muscle cell to depolarize and contract. Disruption at this junction can lead to conditions like myasthenia gravis. Motor pathways are responsible for controlling voluntary and involuntary movement. Key tracts include the lateral corticospinal tract, which carries signals for skilled movements, and the corticobulbar tract, which controls cranial nerve function. Additional tracts such as reticulospinal, vestibulospinal, and rubrospinal modulate posture, balance, and motor tone. Together, these pathways coordinate complex movement. Sensory pathways deliver information from the body to the brain. The posterior column transmits fine touch, vibration, and proprioception. The anterior spinothalamic tract carries crude touch and pressure, while the lateral spinothalamic tract conveys pain and temperature. These tracts help the CNS interpret both internal and external stimuli accurately. The brain and spinal cord are protected by several layers. The cranium is made up of 8 bones that encase and shield the brain. Overlying the skull is the glia aponeurotica, a strong fibrous layer that helps absorb force. The subgaleal space can temporarily store blood during trauma, which helps reduce pressure on the brain. The meninges are 3 protective membranes surrounding the brain and spinal cord. These are the dura mater, arachnoid mater, and pia mater. Between the dura and arachnoid is the subdural space, while the subarachnoid space, located between the arachnoid and pia mater, contains cerebrospinal fluid. These layers provide cushioning and protection. The image likely shows the layers of the meninges and the spaces between them. These structures serve to cushion the CNS, maintain a stable environment, and help circulate cerebrospinal fluid. Meningeal inflammation, as seen in meningitis, can severely disrupt this system. Cerebrospinal fluid is a clear, colorless fluid that provides buoyancy and protection for the brain and spinal cord. It circulates through the ventricles and subarachnoid space. CSF is produced by the choroid plexuses and reabsorbed through arachnoid villi. Normal volume is 125 to 150 ml, constantly replenished to maintain pressure and remove weights. The vertebral column consists of 33 vertebrae divided into cervical, thoracic, lumbar, sacral, and coccygeal segments. Intervertebral discs, with a gel-like nucleus pulposus, absorb shock and provide flexibility. Together, the vertebral column and discs protect the spinal cord while allowing mobility of the trunk and neck. This image likely shows the detailed structure of the vertebral column and intervertebral discs. The nucleus pulposus, located at the center of each disc, absorbs shock during movement. Damage to these discs, such as herniation, can compress spinal nerves and cause pain or neurologic deficits. Here, you may see applied anatomy, such as spinal landmarks for procedures like lumbar puncture or epidural injection. The spinal column's design offers both structural support and accessibility for medical interventions. Understanding this anatomy is essential for safe and effective neurological care. The brain receives about 20% of cardiac output, totaling around 800 to 1000 ml per minute. Blood flow is autoregulated, meaning the brain can maintain constant perfusion despite changes in systemic pressure. Carbon dioxide levels are the primary regulator of cerebral blood flow. The internal carotid and vertebral arteries form the circle of Willis, which provides collateral circulation in case of blockage. Unlike the arteries, cerebral veins do not follow a parallel path and are divided into superficial and deep systems. These veins drain into the venous plexuses and dural sinuses, which eventually empty into the internal jugular veins. Proper positioning is important during head trauma care to prevent venous compression and rising intracranial pressure. This image likely displays the cerebral circulation system, including arteries, veins, and the circle of Willis. This complex vascular network is critical for delivering oxygen and nutrients to brain tissue. Any disruption can lead to strokes or transient ischemic attacks. The blood-brain barrier is a selective filter that protects the brain from harmful substances in the blood. It is formed by tight junctions between endothelial cells and is supported by astrocytes, parasites, and microglia. While it keeps out pathogens and toxins, it also limits drug delivery to the brain. Understanding this barrier is vital for pharmacology and neurologic therapy. Spinal cord blood supply comes from the vertebral arteries, anterior spinal arteries, and posterior spinal arteries. These vessels form a network that ensures continuous perfusion. The aorta also provides segmental arteries that reinforce spinal circulation. Disruption to this supply can cause paralysis or sensory loss. The image likely shows arterial and venous branches supporting the spinal cord. Unlike the brain, the spinal cord's venous system closely mirrors its arterial system. This parallel design supports efficient blood exchange and drainage. The peripheral nervous system includes cranial nerves and spinal nerves. There are 12 pairs of cranial nerves, which arise from the brain and may be sensory, motor, or mixed. The 31 pairs of spinal nerves emerge from the spinal cord and are all mixed nerves, containing both motor and sensory fibers. These nerves form the communication lines between the CNS and the rest of the body. You may see an overview of the cranial nerves and their functions. These nerves control everything from smell and vision to facial movement and heart rate. Mastery of cranial nerve function is crucial in both neurological exams and emergency assessments. Spinal nerves are structured like trees. They begin as rootlets, which merge into roots and then form a nerve trunk. The trunk divides into rami, or branches. The anterior rami form complex nerve plexuses, while the posterior rami serve specific skin areas known as dermatomes. This organization allows coordinated motor and sensory innervation. Illustrations likely show the detailed structure of a spinal nerve and its branching pattern. Understanding how these nerves connect helps in diagnosing nerve injuries and determining their clinical effects, especially in trauma or surgical recovery. The autonomic nervous system regulates involuntary body functions, such as digestion, heart rate, and respiratory rate. It has components in both the CNS and PNS and is divided into two antagonistic branches, sympathetic and parasympathetic. It uses both preganglionic or myelinated and postganglionic or unmyelinated neurons to transmit signals to target organs. The sympathetic nervous system initiates the fight or flight response. It is organized in the thoracolumbar region of the spinal cord, from T1 to L2. Preganglionic neuron synapse and sympathetic ganglia, and postganglionic fibers travel to target organs. This system increases heart rate, raises blood pressure, and mobilizes energy during stress or danger. In contrast, the parasympathetic nervous system promotes rest and digest functions. It originates in the craniosacral regions, cranial nerves, and the sacral spinal cord. Preganglionic neurons travel close to the organs they serve before synapsing with postganglionic neurons. This system lowers heart rate, enhances digestion, and conserves energy. The autonomic nervous system uses two main neurotransmitters. Acetylcholine, involved in cholinergic transmission, is released by sympathetic and parasympathetic preganglionic neurons and parasympathetic postganglionic neurons. Norepinephrine, part of adrenergic transmission, is released by most sympathetic postganglionic fibers. The type of neurotransmitter determines the organ's response. The ANS maintains homeostasis through opposing actions. Sympathetic stimulation prepares the body for emergencies by increasing blood glucose, heart rate, and blood pressure. Parasympathetic stimulation, on the other hand, slows the heart, lowers blood pressure, and enhances digestion and waste elimination. These systems work in balance to regulate the internal environment. With aging, the nervous system undergoes gradual changes. There is a decrease in the number of neurons, along with loss of brain weight and size. Accumulation of lipofuscin and neurofibrillary tangles is common. Neurologic responses slow down, affecting reflexes, coordination, and short-term memory. Understanding these changes is vital in elder care and neurology. This chapter covered the structure and function of the entire nervous system, from neurons and synapses to the brain, spinal cord, peripheral nerves, and autonomic control. It explored how the nervous system is protected, how it communicates, and how it adapts with age. Mastery of these principles forms the foundation for understanding neurological disorders and clinical practice in neuroscience.